Dear students, we are here to discuss about the second chapter, Sexual Reproduction in Flowering Plants. This chapter has very large contents. In this chapter, we are supposed to discuss about pre-fertilization, fertilization and post-fertilization. But here we are supposed to discuss in this segment only about the pollen grain formation. In this series, we will discuss first about the male reproductive unit, stamen, anther development, microsporogenesis, pollen grain formation, development of male gametophyte, pollen viability, and pollen allergy. Now, let us start the topic. Sexual reproduction is the process of development of new organisms from two parents through fusion of male and female gamete. The flower is the main structure concerned with reproduction. The reproductive organ or the sporophylls are produced within the flowers. The sporophylls are of two types, microsporophylls, stamen, and megasporophylls, carpel. Carpel is distinguished as ovary bearing ovule, style, and stigma. Stamen is distinguished as filament, anther, and connective. The whole process of sexual reproduction in flowering plants can be divided into three steps: pre-fertilization, double fertilization, and post-fertilization. Pre-fertilization. The pre-fertilization events can be studied under following points: pollen grain formation, embryo sac formation, pollination, and pollen pistil interaction. Here we are supposed to study detail about the pollen grain formation. First, about the male reproductive unit, stamen. A stamen is the male reproductive unit of angiosperms. It consists of an anther and a filament. The anther is bilobed and the lobe encloses 4 pollen sacs or microsporangia. Each pollen sac contains number of pollen grains. The 4 pollen sacs lie in 4 corners of dithecous anther. Dithecous anther is an anther that contains 2 lobes that are joined together by a non-sporangious tissue called as the connective. The anther wall is made up of 4 layers of cells. An anther dehisces by slits to liberate pollen grains. Anther development. The anther development starts from a mass of homogeneous meristematic cells surrounded by an epidermis. 4 lobes are formed and 4 layers of archisporial cells are differentiated. Archisporial cells, a cell or a group of primitive cells that form cells from which spores are developed. Each archisporial cell divides to form 2 types of cells, a primary parietal cell and a primary sporangious cell. The parietal cell divides into several times to form the anther wall, whereas the sporogenous cell undergoes fewer divisions and forms the microspores or pollen mother cells, PMC. The innermost layer of cell wall that lies in the contact with the PMCs forms the tapetum. The tapetum is significant in pollen development. Tapetum is a tissue present within the anther that provides nourishment to the growing spores. The layer below the epidermis forms the endothecium. Wall layers of anthers: epidermis, endothecium, middle layers, and tapetum. Epidermis, single layer of cells and protective in the function, while endothecium, single layered second walled. Cells have a cellulose thickening with a little pectin and lignin. It helps in anther dehiscence. Middle layers ranges from 1 to 6. The middle layer degenerates at the time of maturity of the anther. Tapetum, innermost layer of anther wall surrounding the sporogenous tissue. Tapetal cells are nutritive. They are multinucleated and polyploid. The UV bodies are deposited in the exine of microspore wall. The tapetum is of two types, secretory or glandular. The tapetal cells remain in situ all through the development of microspore and finally they degenerate. Amoeboid or periplasmoidal, the tapetal cells break through the radial wall to release the protoplast into the pollen chamber. These protoplasts now fuse to form the periplasmodium. Now discussion about the microsporogenesis. Very important question for the board exams. The formation and differentiation of microspore is called microsporogenesis. The pollen mother cells undergo meiosis. Each forms tetrahedral tetrads. Cytokinesis may be successive or simultaneous. Tetrad are of 5 types: tetrahedral, isobilateral, dicuspid, and T-shaped and linear. Tetrahedral is most common. In successive type, the cell wall is formed after meiosis 1 as well as meiosis 2, forming an isobilateral pollen tetrad. It is characteristic feature of monocot. In simultaneous type, each nuclear division in microspore mother cell is followed by cell wall formation. The microspores are separated from the tetrahedral arrangement. They are then surrounded by a two-layered wall. The outer wall is called as the exine and inner wall is called as intine. The pollen grains are the first cells of the male gametophyte. The tapetum gets used up and the anther becomes dry structure. This liberates the pollen by dehiscence of the anther. All the 4 nuclei in a tetrad remain functional to form 4 microspores. In some cases, all the 4 pollens remain attached, forming compound pollen grains, juncus, jatropha. In family Asclepiadaceae and Orchidaceae, the microspores are present as pollinium. Pollinium, it is a mass of pollen grains present at each anther lobe. As the pollinium is attached to the pollinating agents like insects, the entire mass of pollen grains is transferred as a unit. Now about the pollen grain. Pollen grains vary in shapes. It is generally round with size 25 to 30 micrometer. Pollen grain is haploid, unicellular body with single nucleus. It has two-layered wall. Wall or sporoderm consists of 2 layers. Outer layer is thick. It is called as the exine. It is made up of sporopollenin. Inner wall is thin and is called as the intine. It is made up of pectocellulose. The exine is thick and sculptured or smooth. It is cuticularized, and cutin is of a special type called sporopollenin, which is resistant to the chemical and biological decomposition. This preserves the pollen wall for long periods. It also possesses proteins for enzymatic and compatibility reactions. Exine is differentiated into inner indexine and outer ectexine. Ectexine is further divided into inner continuous foot layer, middle discontinuous, baculate layer, and outermost discontinuous tectum. Tectum aids in the identification of pollen grains and assigning them to their family, genus, or species. Pollen grain contains regions of pores or furrows. In these regions, exine is absent. When the areas are circular, they are called germ pores. When the areas are elongated, they are called germ furrows. Intine is thin and elastic. It is made up of cellulose and pectin. During pollen germination, it is the intine that extends out to form the pollen tube. The cytoplasm of the pollen grains is rich in starch and unsaturated oils. They are initially uninucleate and later become 2 to 3 celled. In calotropis and orchids, the pollen of each anther lobe is formed a characteristic mass called pollinium. Pollen grains can be monoclopate or having one germ pore, biclopate two germ pores, and tricolpate three germ pores. Each branch of a study of pollens is called palynology. Now discussion about the development of male gametophyte. The nucleus increases in size. Inside the pollen grain, it divides mitotically to produce two unequaled outer cells, a bigger vegetative cell or tube cell and a smaller generative cell. Pollination can occur when the pollen grain is two-celled tube plus generative or three-celled tube plus two male gametes. However, in plants such as cereals, the male gametes form while the pollen is still within the anther. In those cases when pollen is shed at two-celled stage, the generative cell divides and after pollen has landed on a stigma. The cytoplasm of generative cell does not contain much of stored food material. Fat, starch, and protein granules are present in vegetative cell. Pollen products, pollen food supplements. Pollen grain contains abundant carbohydrates and unsaturated fat. They are used in the form of tablets and syrups for enhancing vital body functions. Pollen consumption increases performance and used by athletes and given to race horses. Pollen creams. Pollen grain protect themselves from ultraviolet rays, thus they are used in creams, emulsions for providing a smoothness and protection to skin. Pollen viability. The period for which Pollen grain remain viable or functional is called pollen viability. It depends upon temperature and humidity. Pollen grains remain viable for 30 minutes. Pollen grain can be cryopreserved in liquid nitrogen temperature at minus 196 degrees Celsius and used as pollen banks. Pollen allergy. Pollen grain produce severe allergy. It causes have fever and common respiratory disorder as asthma, bronchitis. Carrot grass, Parthenium hysterophorus, is major source of pollen allergy. It also causes harm to internal body organs. It was introduced in India along with imported wheat. That's all. Have a nice day.